last lecture. So we talked last lecture about the action potential, the ionic basis of the action potential. We found that it was based on voltage-dependent changes in permeability, where you shift from being permeable to potassium to being very permeable to sodium. The voltage tries to meet that sodium equilibrium potential, and you get a spike. Okay, we learned about this is based on a fast inactivating inward current and a slower outward current. So the depolarization is because of that inward current, and then the outward current is important for the repolarization of the membrane. Okay. So when you go back and look this over, try to really get that distinction between inward and outward current. So an inward current is positive ions flowing into the cell, which happens to be sodium in this case, the typical inward current. And then the outward current is positive ions flowing out of the cell. Okay. And by convention, the inward current is negative going in our plots of current, and the outward current is positive going. Okay. It's just a convention. If I've done it the other way around, just that needs to be clear in your heads. Okay? You will be asked about this. All right, and so basically you have this positive feedback loop because depolarization leads to more opening channels, more opening sodium channels leads to more depolarization, and you get this all or none effect. Okay. So why do we need this all or none effect? And the main reason is that you need to get your signal from point A to point B, from your brain to your toes. Okay, and it needs to be very fast. So how do you get from point A to point B? So the rest of yesterday's last lecture is going to be on the propagation of these impulses. So first we have to understand how passive current flows, passive current propagates in an axon. Okay, we're going to do this by sort of walking through experiments where we have. Let me back up. Oh, I just wanted to make one more note. There were a couple of requests, but there are every time, to have the PowerPoint slides posted prior to the lecture. I'm not going to do that. They're going to be posted. That's class policy after the lecture. I'm sympathetic. If you're having trouble kind of keeping up with the material, because we are covering a lot and going fast, try reading the chapter before the lecture. I know people don't like to do that, but that's probably the best way to kind of keep up with the lecture. And other people, you're welcome to take the lecture, record the lecture, but that's probably the best way to do it. All right, so moving on. Propagation, we're going to look at how current passes through an axon. Okay, and so we're going to first talk about current pulses that are not large enough to generate these action potentials. Remember, we talked a little bit about passive currents before and the charging of the membrane. Okay, so let's perform our experiment where we have an axon, so our squid giant axon, this is big tube, and we're going to stick 2 electrodes in, well, multiple electrodes. So first we're going to put in an electrode here, and this is going to be our stimulating electrode. We're going to inject current into the axon, and that's actually not shown here. And then we're going to record the voltage response. So if we inject and record at the same spot, we see this passive charging up in the membrane. And then we reach that steady state voltage that's defined by Ohm's law, V equals IR. Okay? Now, once you inject current, it has to go somewhere, right? So it typically will flow in both directions of the axon for some distance. But as it flows along, a little bit leaks back out the membrane, right? You've got all of those leak channels. And so there's always a little bit of leakage. And so the signal, which starts off at this level, gets smaller and smaller and smaller and smaller. Because the current, right, currents always have to sum. So the current at this point is the sum of, well, I guess we could subtract. So you could take this current, subtract whatever has leaked out, and that's what's left to charge the current. That's one way to think about it. At any rate, the current that is available to charge the membrane gets smaller and smaller. And we can plot this down here, where the peak membrane potential at different points of recording along the distance along the axis. So this is like we have a whole series of electrodes stuck along the length of the axon. And you can see the decline in the amplitude of the passive response. Okay, this is just another way of plotting it, where you can compare that at the recording site, which is 0, so this is distance from the current injection. So recording the membrane potential at the current injection, you get this. If you move 0.5 millimeters away, it's smaller, but you'll notice it's also slower, right? And there's a little bit of a delay. So things are getting slower, things are getting smaller as you move away from the point of stimulus. Okay. And this is because this injected current leaks across the membrane, and it leaks more and more, so you lose more and more as you move along the membrane. Okay, so we can come up with a mathematical description of this. This is an exponential function. And so this is a function of the initial voltage, V0, and then it exponentially declines with the distance along the axon and the exponent, and then divided by this kind of funny number, this lambda. Okay, and this lambda is kind of an interesting thing. So this is a way to kind of define just how far does that passive signal propagate. And so given the properties of an axon, you can calculate the lambda, which is considered, a life constant. Okay, so there's one lambda is the distance that the signal propagates before it loses roughly 2/3 of its amplitude. Okay. Okay, and in a squid, in the giant axon, this lambda is about a millimeter. Okay, but in mammalian brains, this can be much, much smaller. Basically, there's a lot of leakage as you go along. And we're going to talk about ways to adjust for that so that the brain can send long-distance signals. All right. Okay, this is just restating what we just said. It's very leaky. Let's see how, what can we do about this space constant, this length constant? Okay, so here's the same equation. Okay, and we can define or calculate lambda based on a couple of properties of the membrane. So remember that in a cell, you have the resistance, the membrane resistance, and that's basically a measure of the leakiness across the membrane, right? So you've got those leak channels. And that's usually called Rm. So that's the leakiness across the membrane. But we can also define the resistance axially along the axon, right? And so that's Ra. And so the space constant is, the square of the space constant is proportional to the ratio of those two. So making one bigger will change the space constant, okay? The other parameter here is this diameter. So the width of the axon is quite important, right? Because the ratio of those two things, if you think about a circle, right? You've got the ratio of the circumference of the circle relative to the area. And as the circle radius changes, those two things don't change the same way, right? Area is a square of the radius. Circumference is a linear of the radius. So as you make something bigger, you change the area more than you change the circumference, right? Everybody remembers their geometry a little bit, maybe? Okay, so what happens is that if you increase the diameter, or the radius, you can alter the space constant. So basically what you have, if you have a really fat axon, like you do with that squid giant axon, the ratio of these two really changes. The Ra gets much, much bigger, right? Okay. The diameter gets much, much bigger, and that makes the space constant bigger. Sorry, these are constants, because these are in sort of resistance per patch of membrane. Okay, so these two are constant, and if D increases, then the space constant increases. So fatter axons, you have more current flow going down the axial component relative to less leakage going out. Does that make sense to everybody? You can kind of think of it as a wire, right? So you have current traveling down a wire. If you've got a narrow little wire, you can only have so much current traveling down. If you make this wire really fat, then you can get a lot of current down the wire. Okay, so that's why you have a switch on an axon. Okay, so now let's do this experiment with, yes, question.
How did RM, or how does it increase with insulation if it's a value of leakiness?
We'll get to that. We'll get to that in just a minute. Well, why don't you, can you answer that? What do you think would
I
happen?
figured if you insulated it would decrease, unless it's inversely proportional.
So the question was, how does insulation affect all this? We're going to talk about myelination. And yeah, basically that's it. Yeah, if you insulate, then you limit the leakiness of the current. And that means the signal can propagate properly. That's exactly right. It's very intuitive. Okay, so what are we going to do now? We're going to look at this experiment. We're going to do this experiment when we give it a bigger pulse, and so now we have an action potential. Okay, so now we're going to do the same thing. Stimulate, record. At the site of the recording, we have an action potential. But now, as we record along the axon, we notice that we always get an action potential, and it's always basically the same height. Okay, so this is plotting the peak of the action potential, and this is this boring straight line. Okay, so this action potential is basically traveling down the axon relatively unchanged. It's an all or none signal, 0 or 1, digital. Okay, but there is a little bit of a delay, right? So the action potential farther down the axon is a little bit later than that. Okay. Yeah?
What do you mean by delay?
So if we define 0 as the time of the stimulus, the action potential occurs at 0 here. But if you look down here, it's around 5 milliseconds later.
Why is there a delay like that?
Why is there a delay? Why is there a delay? Because it's traveling. Because it's a traveling signal. It's not instantaneous. And we're gonna get to that. Okay, so propagation. Let's talk about propagation of action potentials, because it's really a fundamentally different thing than the passive movement of voltage of the membrane potential changes down the axon. Okay, so how are we able to get these action potentials? Okay, two of the clues is that the amplitude of the action potentials is always the same, it's constant. And so we have this all or none behavior. And the action potentials occur later and later as you move down. And so there's a measurable rate of transmission. And we call this a conduction delay. And we'll come up with that a little bit. So this all or none behavior. Do you remember what causes that all or none behavior?
Graded potential?
The graded potential, no.
The inactivation of sodium.
The inactivation of sodium. A little before that. What happens right before that?
The sodium voltage gated channel.
The sodium voltage gated channel. What do they do to the membrane potential?
They make it more positive.
They make it more positive, which causes what? Depolarization, which causes what?
Inactivation.
Yes, but first we'll say more activation. So we're going to call that the positive feedback cycle. So very quickly. And then you get to the inactivation. That happens too. Okay, but this all or none behavior gives us a clue as to what's going on with propagation of the action potentials. Okay, and what's going on is that you're having basically a chain reaction of that sodium channel positive feedback, right? Okay, so let's look at what's happening. So we stimulate here. So here we have our axon, and now we've inserted these channels to kind of look at what's going on. So we have sodium channels, and we also have potassium channels for good measure. Okay, but let's look at the sodium channels first. So we stimulate, sodium channels open, you get an influx of sodium, a depolarization, and that causes this signal to propagate down the axon. Okay, so initially, here's at time 1, at point A, which is here, we have an action potential. Okay, it's all very good. And this signal is propagating down the axon. Okay, and it's declining as it goes along, let's say. And so at point B, eventually, the signal is such that you have openings of these sodium channels. A little further down the axon, everything happens again. You have influx of sodium, depolarization, and then that boosts the signal. Okay, Okay, so now you have an all or none signal at this point, and here's your action potential at point B. It's a little bit later because it had to wait to charge up because it had to get the signal that flows down the axon. There's sort of a finite time there. Okay, so now we have an all or none signal, and again we have current flow. And notice it's actually flowing in both directions. Okay, but let's look in this direction first. Okay, so what happens? Okay, so this flows down, and then the whole thing happens again. Let me come back to this in just a minute. So at point C, we have the whole thing happening again. Sodium channels open, influx, depolarization, and boom, we have a spike. Okay, so it's the flow of current down the axon that causes that delay. Okay, but you have this sort of chain reaction as you're going down the axon. So let me back up for just a minute. Oops, other way. Okay, so current is actually flowing in both directions, right? So at the point of your original action potential, what's happening here? Here you have, initially you have that sodium current, but then that's immediately followed by a potassium current. So you have potassium channels that open, and they're pushing the membrane in the other direction. At the same time, you also have sodium channels inactivating. So that little chunk of membrane actually becomes what we call refractory for those two reasons. You have potassium currents that are pushing the membrane away from our threshold, and you have sodium channels that, because they all just opened, they go into an inactivated state and they're not available anymore. Okay. And so this is what we call our refractory period. This is an inactive state, and it's not going to fire an action potential again at that point. Does that make sense? And this is actually quite important. It really sort of limits the ability of the neuron to fire at sort of infinitely high rates. All right. So refractory period. Let me just recap. This depolarization that opens all of these sodium channels, and the slower activation of the potassium channels leads to the repolarization of the membrane as the action potential sweeps along that length of the axon. Sodium channels are inactivated, potassium channels are activated for a brief time, and you basically kind of have to wait for everything to get back to normal before you can fire another action potential. Okay, another way of looking at it is having all of those channels open means that the resistance of the membrane is low. And so we need more current to depolarize the membrane. Okay, so with R is small, you need a lot more current in order to get the same voltage, right? And that's where that sort of some of that leakiness of the membrane comes from. Okay, and so that's our refractory period. Okay, as I said, this limits the number of action potentials that you can produce. And different neurons sort of regulate this different ways. Some neurons fire really fast, and other neurons kind of slow things down for different reasons. Okay, and another explanation is this is why action potentials don't propagate back toward the point of their initiation. They're kind of unidirectional, right? So they're not, you don't get into this cycle where you're kind of bouncing your signal back and forth all the time.
If you're talking about the RM being low on the last slide,
Yeah.
you're saying the RM, the leakiness of the channels are less leaky because of the inactivation. Is that what that means?
Right. So you have, sorry, it's the potassium channel. So you have lots of potassium channels open, and that makes the membrane very leaky. So you can think of it as low resistance is high conductance. So there's a lot of open channels that happen to be the potassium channels. The sodium channels are closed, right? Those are inactivated. So those aren't contributing. The potassium channels are open.
Oh, so low RM means it's more leaky.
Yes.
Oh, okay.
Yeah, that's always a point of confusion. So you're saying low RM means more leaky. So flip it over in your head and say low RM is large conductance, which is lots of open channels. Yes?
Yeah, Google suggested it's resistance to leakiness. That's the, like, RM is the resistance to leakiness. So when it's flow.
Yeah, you know, come up with your own
Which makes sense.
way of thinking about that. I think electrical engineers have a good feeling. I can think of it as, you know, if you have a wire and it's resistant to electrons flowing. If it has low resistance, it says, oh, you know, just keep going by. Okay. Resistance is futile.
Thank you.
All right. Yeah, I mean that's a very common point. But I still, I paused there for a second. Right? Still do it. All right. Moving on. Okay, so we talked about that delay. And so there's a conduction speed that happens. Okay, so what we're talking about for the squid is pretty slow. It's like 2 meters per second is the propagation speed. Okay, and so we need to have faster speeds if we want to react quickly. And so the squid kind of does it by making these really big axons. That's not really a practical solution for a lot of animals. Certainly not for all of your nerves. And so another solution is to wrap it in myelin. So remember we talked about oligodendrocytes and Schwann cells that do the myelin wrapping. And so you have this insulation. So just like a wire, you can wrap it with insulation to improve its function. Okay. So just as was said, this changes the RM, so we can get less leakage of the passive current. Okay, so then what does that do to the signal, right? Because we were talking about you need to have this sodium channel regenerative mechanism in order to propagate. Okay, so the solution that the nervous system has come up with is to have the best of both worlds. So we're going to wrap certain sections in myelin, and then every once in a while we're going to have a gap. Okay, and at this gap is where we're going to put our sodium channels. Okay, so you initialize a signal here, and then it flows along the axon to the next point, and then causes those sodium channels to open, which sends the signal along, and so on. Okay, and this is often referred to as saltatory action potential, which basically is, I forget if it's Latin or Greek, but what it means is sort of leaping signal propagation. So let's look at this a little bit more. So it's a little counterintuitive as to how that results in a faster signal propagation. It certainly maintains the signal, but it's actually faster, and let me explain why. Okay, so here's an example of how the action potential sort of jumps from point to point. So we have point A, B, and C, and we have an action potential, and there's still a little bit of a delay there. Okay, but let's compare an unmyelinated axon and a myelinated axon. And why is the signal faster in a myelinated axon? The reason is this. Okay, let's see if we can work through this. So in the unmyelinated axon, we talked about, we initialize a signal, and then there's a passive decay of the potential as some of it leaks out. And so you have to sort of imagine that, let me see if I can write this. I know it's dark from the back. So let's imagine that this is your initial signal, and it's declining like that. Okay. So if we wrap it in myelin, what's going to happen to this signal? Is it going to be bigger or decay faster? Bigger. So start off the same, but because it's less leaky, it's going to move along like that. So at a given point, you've got more of the signal maintained, right? Okay. So what that means is that, let's see, so if your threshold for firing an action potential is, yay high, the myelinated axon is going to meet that threshold when the unmyelinated axon is not. Okay? So you're going to maintain the signal And when you get to this point, okay, you have enough signal to fire these sodium channels, whereas in the unmyelinated case, because of the decay of the signal, only these are going to be able to fire. These haven't seen enough signal to fire just yet. Yes?
So, sorry, you can go first. So for the top, is it kind of, for the unmyelinated, is it kind of like there's so much signal leaving that it's gonna take, well, okay, for the myelinated, is it saying that you've got a lot of signal and it's staying that way, so it doesn't need as much of the extra to get it to reach the action potential compared to the unmyelinated where there's a lot of things, so you need like a lot more for your celebration.
I didn't quite follow. I would repeat it, but I didn't quite follow it. But let me back up and maybe take another run at it. Okay.
Yeah. It's
So
that's a pressure cooker and that is losing its pressure.
yeah. So the comment is it's a pressure cooker and it's kind of losing its pressure. I think you should imagine that the signal in this stretch of axon looks like this. Okay, so if we have a threshold, for activating sodium channels, right? In the unmyelinated case, you have to be really close to the point of origin. In the myelinated case, you can be way out here, and you have enough signal to fire another action potential. Right? Does that make a little more sense? Okay, so these are seeing a strong signal. So at the same time point, these, just a second, these channels are going to see a strong enough signal to fire, whereas the only channels that are going to see a strong enough signal are back here. So there's a space difference there. Yeah.
So would it be fair to say that the myelinated one would travel faster because it would need to trigger fewer action potentials to go the same distance?
Yeah, it's not really fewer, it's that you maintain so much of the signal itself propagates faster.
Faster. So like in order to cover the same distance, would you end up getting fewer of those action potentials because like each one can go a further distance?
Say that again?
So would each like action potential peak go a further distance before it needs to, I guess, like trigger another one to propagate the signal?
Kind of, yeah. So the, let me see if I can repeat what you said for the people in the back. So the peak of the action potential would go a farther distance in order to propagate the next thing. Is that kind of, yeah, I would say that, yeah. Yeah, so the peak of this sort of traveling wave, it's not, the boost in the traveling wave is happening farther to the left here than in the myelinated case. And so you've actually kind of pushed the signal ahead a little bit because you're not leaking as much.
Do myelinated axons have less channels because like they can't trigger during the insulation?
Do myelinated axons have fewer channels because you don't need to propagate sort of continuously along the membrane? And I actually don't know the answer to that question. Logically, I would say yes. So in that sense, this might be a more efficient way of doing it. So let's kind of walk through this whole thing again. Okay, so in an unmyelinated axon, you have a signal that propagates. In a myelinated axon, you have a signal that stays larger, farther. The next boosting activation in a myelinated axon happens here. So you get a boost in the signal, and then that travels along. In this case, the boosting happens a little less far along the axon, and then the signal travels a little less far. And then at the next jump, you have activation here. So this is all at the same time. So you have activation at this point, and in the unmyelinated axon, you're only getting activation at this point. Okay, so if you think of this as, let's I'm going to flip through these like a little animation. Okay, so if you look at it as sort of a race between those two signals, you can see it's getting farther along the myelinated axon than the unmyelinated. Okay, I'm sure this is a little annoying for you guys, but you kind of get that feeling. Things are traversing, and it's traversing faster along the myelinated axon because you're losing less of the signal. All right.
Sorry.
Yes.
In addition to having faster speed, is the myelinated axon more likely to have action potentials at all compared to the unmyelinated?
Yeah. So the question is, are myelinated axons more likely to fire an action potential than an unmyelinated action? Another way of saying that is, do unmyelinated axons basically fail at some point? And I would say yes, they probably do. Because anytime you're losing that signal, you're risking dropping below that threshold level and sort of terminating that positive feedback. So yeah, I would say yes.
Would that fail be considered just a graded potential instead of an action potential?
So that's an interesting question. So once it gets below a certain threshold, yeah, basically it becomes a graded potential. And then it could be treated like any other graded potential. Yeah, that's a good way.
Thank you.
All right, so to summarize, so we've talked about the action potential propagation requiring both these passive and these active mechanisms. And you can increase the speed by improving the passive current flow. I realize this is a lot of text, but this is basically what we've just talked about. So you have this passive current flow, and by increasing the diameter of the axon, you can get more flow in the axial direction relative to the leak out the membrane. Okay, and then insulating the axons makes them less leaky. Okay, and so that improves that passive flow. And basically this is why we have myelin, is basically to improve that process. And then you have this really cool trick of spacing your booster units, those nodes of Ranvier, in between the myelin in order to basically get a faster signal. All right. And okay, I'm going to kind of skip this. This is basically everything that we just talked about in the last lecture. So now it's 11:30 and we finally finished the last lecture. So I'm just going to skip this and come back to it in minutes. And there's a couple of good, if you go to the textbook website, you should be able to look at the animations that are there. If anybody has trouble accessing those, I can probably post them on Canvas. I don't know, I haven't done that before, but you can probably do that. And it'll walk you through the voltage plan and axon conductance. Okay, and then here are some review questions. You can look at these at your leisure. We need to move on to today's lecture. So now we're going to continue talking about ion channels, but more in terms of what ion channels are really about. Okay, so we talked about the action potential. Okay, so this is basically just summarizing what we just did. So we know that they're voltage dependent. Ion channels are voltage dependent. We know that they're selectively permeable. They choose only sodium or potassium. We're going to look, there's more different kinds of channels. They depend on the electrical chemical gradient. And so what are the mechanisms for creating this? We know how electrical chemical gradients come about, but now we're going to talk a little bit about permeability and voltage dependence. And trying to really understand ion channels. The switch ion axon was a great leap forward. But now we want to understand these on a more sort of mechanistic level. And so we're going to use a new technique in order to really look at individual ion channels instead of overall currents. Okay, so we're going to look at ion channels and their voltage sensitivity. We're going to focus on these voltage dependent channels. We'll talk a little bit about the pumps. We talked about these a little bit before, that maintain the chemical gradients. And then there are other pumps that are around that, you know, really shuffle ions around so that the cell can work. Anybody have another ion that's really important to control concentrations of?
Calcium.
Calcium, right? So all of your physiology is hugely dependent on calcium. So there's lots of different examples of this. And they're placed strategically in the cell in order to get the gradients in the right way. Okay, so how are we going to look at individual channels? And this really depended on the development of what we call patch clamp recording, which was really kind of a collection of modes of recording. But they're all kind of dependent on the same core principle. And that is that we're going to use a micropipette, so like a little glass capillary, that is going to have a very blunt end that then sucks onto the cell membrane. Okay, so here's kind of a blow up of this region. And so it's really going to attach very tightly onto that membrane. And presumably that membrane contains the ion channels that we're interested in. This was developed by Bert Stockman and Erwin Mayer. And they got a Nobel Prize for this, but pretty much now, you know, everybody does this. So it's a very, very common technique. And the idea that this was in order to record from, you know, what they call small cells, but sort of small relative to squid axons. But here's an example. Oh, so this is a photo of an actual patch clamp recording. This is from my laboratory. So there's a neuron. You sort of see the outline of a neuron and a brain slice, and you can kind of make out the pipette. And you can actually sort of see the pipette bump up against the membrane and suck onto the membrane. Okay, so once you've sucked onto the membrane, and once you've done that, there's almost no ion flow between, you know, leaking out. So you've got a really tight junction between the glass and the membrane. Okay, and then you can do a number of things. You can either rupture the membrane, and so now you have electrical continuity. You have a wire in the pipette and salt solution in the pipette, and then you can record the voltage inside the cell, just like if you took a sharp electrode, kind of like we talked about before. Okay. But there's a couple of other really useful techniques where you basically pull off a little patch of membrane. Okay, so the membrane is still kind of covering the mouth of this pipette, and now your recording is entirely dependent on the channels within that little chunk of membrane. Okay, and you can kind of do a little trick where you can sort of flip the membrane inside out. And so this is called an inside-out recording. So the inside of the cell membrane is exposed to the extracellular medium. And here, this is the outside-out recording. And so what happens is you rupture, so you rupture the membrane, so there's a hole here, and then you pull off a chunk of this membrane, which then reseals. And now the outside of the ion channel is flipped upside down, and so now the outside of the ion channel is exposed to the extracellular medium. And so this is really useful, especially for ligand-gated channels. So you have access to the outside. Okay, and so these different modes of technique are really useful for asking different kinds of questions. So if you use the whole cell mode, you can study the cell properties of the whole cell, just as if you had stuck in an electrode in the middle of the cell. And in particular, you can study synaptic inputs and firing patterns. An outside-out patch, the inside-out patch means that you can study detailed study of ion channels, because you can modulate the intracellular domain. So you can imagine channels that are being modified by intracellular factors, and so you can control that from the outside, wash things on or off. And then the inverse, the outside out, you can study ion channels, in particular ligand-gated things, because you can apply the ligand to the outside. Okay, and so this is sort of a typical patch clamp rig. It's very difficult to see much, but the electrode is right here, and your little brain slice is right in there, and you can see it through the microscope. Okay, so let's talk about sort of a specific experiment here. And we're still talking about the Hodgkin-Huxley channels. What really underlies those currents? And can we get more detail on these channels? Okay, so we're going to record from, in this case, this is an outside-out patch. Okay, and we're going to block potassium channels. So we'll use pharmacology to isolate the sodium channels from a patch of that sweet axon. Okay, and let's perform an experiment. We're going to do our usual. Because we've got such a teeny tiny little patch of membrane, it's very easy to control the voltage. It's basically that that compartment experiment, except it's very small, right? So you can control the inside, the voltage across the inside to the outside. And so we can control the membrane potential. We'll go from, say, -80 up to, I don't know, what's that, -20 or so with a step. And then measure what the currents look like. Okay, so here's the current. So each of these lines here is a single trace. So you have time on the bottom, and this matches the time of the step up here. And then these are just multiple examples and from different trials. Okay. Okay, so we know that these are sodium channels because we can throw on TTX and it blocks. Okay? But there are some really major differences between these currents and the currents that we were looking at last lecture. Does anybody notice anything in particular? Got lots of hints here. What's different? How big are they? They're teeny tiny. They're very small. So there's a couple of picoamps. Okay, and before we were talking about, what was it, maybe nanoamps of current before. Okay, so they're very small. They're also shaped kind of funny, right? They're a little bit sort of squarish. They kind of come on and go off. And they're also a little bit random, right? So you have the timing of the onset is kind of slightly random. Sometimes they stay on a little longer than others. Sometimes they look like they kind of come on and close and come on and close and flicker around. So they're a little stochastic, okay? But you'll notice that most of these openings happen very early on. So what does that remind you of with the sodium current? What did you know about the sodium current? It happens early on. There's a fast onset, and it turns off, right? Okay, so that seems kind of consistent. Okay, so these are very small, but even though these are small, these represent a lot of ion flowing, and they're all basically the same amplitude. If you go out and measure them, so they basically come on, have a certain amplitude, and then shut off. Okay, and it's the unitary amplitude that kind of gave people a clue. It's like, well, maybe this is just a single channel opening and closing. That's kind of cool. Okay, so you have this elementary conduction unit, and this is what makes this a single channel recording. Okay, so let's take a whole bunch of those individual microscopic currents and average them out, and you get something that looks like this. There's sort of an early onset and then an offset, which reminds us a lot of the macroscopic current that we recorded from the whole axon, where you have a rapid onset and then an inactivation of the current. Okay, so there's a real match in the kinetics. If you plot the voltage dependence, so here this is the probability of channel opening versus the membrane potential, that also matches. So we have a really close correlation between the properties of these two things. And so we can kind of infer that they're really the same thing, right? So we can ask a bunch of questions. Do the currents reverse at the expected potential to carry sodium? We didn't show that, but yes, you would look for that. So you would hold at different voltages and see a sign inversion. Okay, do the kinetics of the opening and closing match? Yeah, you saw that. Okay, do they have the same voltage dependence? Yes, they both activate around -30 to -40. You can define the half inactivation. You can fit a curve to it. And do they have the same pharmacological profile? Yes, they're both blocked by tetrodotoxin. So we can kind of infer that these are one and the same thing. Okay, so we can do the same thing all over again with potassium currents. Now we're going to use TTX to block the sodium, apply a step, and measure these currents. So how are these different from the last one?
The channels open later than the sodium
The channels, maybe the channels open a little later. What else? For a longer period, yeah. So they go on and they stay on. Do you notice anything about the direction? Yeah, the polarity is different. So these go up and the other ones went down. Right? So that means you have ion flow in the opposite direction. And we all know that potassium ions and sodium ions flow in opposite directions. Okay, so we can basically go through the same logic and say, okay, if we add them all up, all those microscopic currents, we get something that looks like this. And it looks very similar to the macroscopic current. It has very similar voltage dependence and so on. Okay, so these are all the same questions that we've answered all pretty much the same way. Okay, so you gotta do this kind of matching. Okay, so let's start to talk a little bit about some of these properties. So we're going to talk first a little bit about the kinetics. So they're voltage dependent. And so what's actually happening with the ion channel? Okay, so what's happening is that the ion channel has different states. So you all learned about proteins and how they can have conformational changes and so on. So this is a very cartoony version of that. So initially, sodium channels are closed. When they detect a change in the voltage, there's a conformational change that opens them, and they have a pore that allows the ions to flow. At some point, there's this little gate down here. And so this is what makes the sodium channels a little bit peculiar, is you have this gate that closes, and it doesn't allow any more ion flow. So even though this pore is open, so you see these charges up here, that kind of implies that it's still in that sort of open pore is still in that open state, but now we've got this gate. And so this is this inactivated state. And so no matter what the voltage is, it's not going to allow ion channels to flow anymore. And then eventually, when you release the voltage, you go back into a closed state. And when it's in a closed state, this little gate flips back open, and now it's ready to be activated. Okay, similarly, for the potassium channels, you have a closed state. And then initially it's still closed, so it opens a little bit later. Opens, allows potassium to flow, stays open, and then it's only when you shut off the voltage or repolarize the voltage that it goes back into this closed state. Yes?
So when we were talking, I was reading in a textbook that when it goes up and down like that, and it's when the voltage goes up and down like that in the action potential, and it's positive, it talked about how even though it was positive and it was going up and down. Is that the reason that it has the gate instead of just going back into a closed conformation? I'm not taking it right.
Maybe we'll come back to that. You can mull over your question. Okay. Well, we can come up with a state model. Smart people can come up with a state model for these channels. So you can come up with this little diagram that you go from closed to open, open to inactivated, and then inactivated back to closed. And part of the implication is that you never go from inactive to open. Potassium channels are a little bit simpler, that you just go back and forth between these open and closed states. And all of these transitions are voltage dependent. It's actually much more complicated than that. Okay, so this is just an example. Do not memorize this little thing. I don't even know what this is from. But the basic idea is that you can have lots of different states, open and closed, and you have all these little rate constants. And so basically it's a kinetic model. And these have been very, very successful in explaining the behavior of these channels. But don't memorize this. Just an example. Okay, so moving on, we have these two classes of channels. They're both voltage gated, and so they must have something in the protein that senses that voltage, right? Okay, Okay, you can probably imagine all kinds of things. And so the fact that these are dissimilar in their channel kinetics, where the sodium channels inactivate, the potassium channels do not, implies that there is some difference in the structure of these channels. So we're going to talk a little bit about channel structure now. Okay, so features of ion channels. There's the ion selectivity. That defines the permeability. Okay, voltage sensitivity and the kinetics, which we just talked about. And most of this can be understood from the structure. So you all know that proteins are pretty complicated natural molecules. You know, we can have the gene, and you can infer from the gene what the amino acid structure is, and then we go from that to secondary structures. It's like, oh, okay, well, here's a helix, and you know, this is hydrophobic and that's hydrophilic. But of course, getting from that to the tertiary structure of how the whole protein fits together is kind of a really difficult problem. And it's hard to come up with that just from the amino acid sequence itself, right? It's a hard problem. So you've got all of those degrees of freedom. Okay, so we really depended on crystallographic studies, X-ray crystallography, to understand a little bit more about these channels. And we'll come back to that in a little bit. Okay, but to study these channels, first of all, we need to express them. To understand their function, we have to express them. So often what you need is what we call a heterologous, pronounce that right, heterologous expression system, which just means taking mRNA from one animal or a known mRNA and expressing it in a convenient system. Okay, so one convenient system, you've probably all seen this before. How many of you have seen this before? Xanthus oocytes. Kind of sounds familiar, looks a little like the moon. And so the basic idea is that this is a system. These are frog eggs from the Xanthus flavus frog, which are really ugly little frogs, but they're very useful. And they lay these enormous eggs that are a millimeter across. These are huge. Okay, and what you can do is you can inject mRNA directly into these eggs, and the egg doesn't know any better. It just produces just a protein-producing machine. So it just produces enormous amounts of the channel that you're interested in. And it's not real picky, so it just transports those channels to the membrane, and then you can record them. It's kind of a cool system. I actually worked with this a little bit as a graduate student. It's kind of a really fun system. The frogs are really fun because they, you know, when they have eggs, you just sort of pick them up and you just sort of squeeze the eggs out of them. All these eggs pop out. It's lots of fun. Poor frogs. Okay, so you can synthesize these proteins, and you can make any protein you want, right? So you can just design your mRNA, substitute amino acids wherever you want. You can truncate it. You can stick different things together, and it will basically produce this protein and stick it in the membrane. Okay, so now you can do patch clamp studies, or one of the reasons people originally used these is because you could actually stick 2 electrodes into the same cell. There are not very many cells in the world where you can take a sharp electrode and stick 2 of them into the same cell. Okay, and that's one of them. So that's sort of 2 electrode voltage lamp. It sort of predated all of the patch clamp. Okay, but the principle is the same, that we're going to control the membrane potential to different steps. So this is a series of steps. So this is, you know, 1, 2, 3, 4, 5, 6, 7, whatever, 7 or 8 steps done individually. And they're just plotted one on top of each other just for space considerations. Okay, but you should consider each of these sort of a separate trial. Okay? And then for each one of these steps, there is a current that occurs with each of those steps. Okay, so you have matching steps here in the resulting current. Okay, and so you can learn a lot about what the function of these proteins are using this methodology. And so over the years, you know, people have come up with functionally a whole bunch of different classes, and then genetically a whole bunch of other classes. And a lot of like the last 20 or 30 years has been kind of trying to put these things together. And it's not as easy as it seems. Okay, but we can come up with these major classes of ion channels. We have our voltage-gated channels, our ligand-gated channels that we'll talk a lot about later on when we talk about synapses. Stretch and heat-activated channels. This is kind of a really popular area of study. These are the trip channels. And you can end up with over 100 known potassium channels. So there's just a tremendous diversity. And what this allows you to do, it allows you to fine-tune the electrical properties of these neurons. And you can get just an enormous array of behaviors from these neurons. So it's great. Okay, and then on top of that, you can have these post-translational modifications and heterologous recombinations, and all kinds of variations on this. Yes?
Yeah, the 100 known potassium channel genes, are those mostly genes that act on the potassium channels or like modified in some way?
Those are the potassium channels.
Like literally there's 100 copies.
Literally, those are the proteins that make the channels that fit in the membrane. Yeah.
So is that 100 different types of genes or 100 different types of channels?
I would, let's see, how do I answer that? It's kind of a hard thing to answer because you can have a gene that has multiple components, that sometimes you use one component and sometimes you use another component. So is that one gene? Is that two genes? Certainly two channels, but it's kind of one gene. So let's call it genes. And of course, it also depends, which animal are we talking about, and so let's put it, just get the order of magnitude right. Hundreds of genes, on the order of 100 genes or so. And then you have all of these variations and subunit combinations that we'll talk about in a minute. And all of these have a characteristic structure, and so on. Okay, so let's talk. We're going to talk mostly about the voltage-gated channels. We talked a lot about sodium and potassium, and then there's also calcium channels, and chloride channels, which we're not even going to get to today. Okay, all these different classes. And these are very selectively permeable to these particular ions. Okay. On the other hand, one difference between these and the ligand-gated channels, besides being the fact that these open only when they have the right ligand, the ligand-gated channels tend to be a little less selective, and we'll talk about that a lot more when we talk about synapses. Because it has consequences for the electrical properties of these channels. Okay, and we're not even going to talk about stretch channels today. All right, so this is all in your textbook. Okay, so just to kind of compare these different classes, we have a sort of limited number of sodium voltage-gated channels that all form a core with a voltage sensor. There's a rapidly inactivating one, but there's also another one that doesn't really inactivate, that has sort of a slightly different function that people are still kind of trying to figure out. Okay, and then you have calcium channels that are actually fairly similar to the sodium channels. Again, there's maybe a dozen genes or so. They're also found heavily in the heart, right? So you have all those cardiac cells that have calcium and so on. And they vary in their inactivation and activation. And they're really important for regulating intracellular processes. Then potassium, we just talked about, that also vary a lot in their activation and inactivation. So we talked a lot about a particular potassium channel that is non-inactivating, but there are also ones that inactivate and have different kinetics to them. And we'll look at a few examples. Then there's the potassium channels that generate the resting potential that are not voltage dependent. Okay. And we'll talk about this a little bit more. Let's get to this in a minute, about the gene structure. So why are these potassium channels? How do you get this diversity of potassium channels? Okay, and I'm going to have a slight diversion. So we talked about tetrodotoxin as a neurotoxin from the fugu, and it's important because it selectively blocks those sodium channels. Well, there's just lots and lots of toxins that come from lots and lots of really fascinating animals that block lots and lots of these ion channels, and a lot of ligand-gated channels as well. Okay, and I think this is kind of a fascinating thing. So basically, you've had this evolutionary experiment that has come up with all of these sort of drug discovery mechanisms to block ion channels. And so let's see this. Here's another thing here. The list just goes on. And these are all really cool animals. In particular, we can talk about, say, conotoxin, which is a calcium channel blocker. And so this was something that was discovered by this guy. Conotoxin is found in these cone snails, which are these predatory marine snails. Don't step on one, they will hurt you. But the idea is that you can use this particular toxin because they're very, very specific. And then you can use them in various medical applications. In particular, this one's being used as, a potential painkiller. But there's lots and lots of applications. And one of the reasons to go yay biodiversity, because basically nature has already done the drug discovery for us. Okay, so if we conduct our experiments on all these different kinds of channels, we can look at their property differences. So again, we're going to do a voltage clamp experiment where we have a negative going step and a positive going step. And here is our channel with lacking in activation. So this is a KB 2.1. So there's a whole range of voltage-gated potassium channels. If you hyperpolarize, there's no current. If you depolarize, there's a long-lasting, slowly activating current, non-inactivating current that has an activation curve that looks like this. This is the voltage dependence. But then there's another one. This is KB 4.1. You don't have to remember these exact channels, but just know that there's a range of different responses. So this one activates when it's depolarized and then inactivates. And so in essence, it's very similar to the sodium channel. So, I don't want you to get kind of too stuck on the sodium inactivates and potassium never inactivates, because there's really a real variety in the responses. Okay, and then there's like a whole list of examples in the textbook just to give you an idea of what the different responses are. Okay, and so these are the examples similar to the squid non-inactivating one, and an example of a potassium channel that inactivates, similar to sodium. Okay, so now let's talk about the basic channel structure. We know this from genetic studies. You can decode the gene, look at the amino acid sequence, determine the secondary structure, kind of infer what the tertiary structure is, and try to come up with an idea of how this thing is being put together. Okay, and typically, this is our little cartoon of a channel, and it's extremely simplified. That if you look at the protein structure, so this is for a sodium channel, you can infer how this protein goes in and out of the membrane. So you have these transmembrane domains. These tend to be helices that have the right, hydrophobicity, so they go in the membrane, and they tend to have sort of 4 big domains here. Each of these are composed of 6 transmembrane or membrane spanning regions. Okay, so you have 6 and 4, and then they kind of come together to form an entire channel. Okay, and most of these you refer to the main. So most of these we refer to the main core forming channel as the alpha subunit, but there are also these what we call auxiliary subunits. So this is pointing to a little beta subunit. So this is a protein that has 1 transmembrane domain. There's another one over here, and these get together with the alpha subunit and form a complete protein, but they really have more of a modulatory effect on the channel. So here's kind of a bigger blow up where you can see these transmembrane domains right here, and then you can sort of infer which one of these sort of fit together to form the core region, and so on. Okay, the sodium channel and the potassium channel, the sodium channels and the calcium channels are quite similar. So this is a calcium channel. Again, we have sort of these 4 big chunks, these 4 big domains, and then each of those have 6 membrane spanning regions. So all together forming the core of the ion channel. Okay, potassium channels are a little different. So again, sort of your standard voltage-gated potassium channel has these transmembrane domains, but now there's just one of them. Okay, and so you actually have to get 4 or 6, some people argue 5 of these individual proteins together to form a functional channel, because you need them to form that pore region. And so they bind together and they sit in the channel together. Okay, but then there's a number of varieties. The calcium activated potassium channel, what's called an inward rectifier. We're not really going to talk about that. This two-pore channel is the one that was sort of recently identified as the leak channel. Okay, it tends to be not voltage sensitive. Okay. And then just to give you an idea, we talked about these families of channels. So you can analyze parts of their structure. This is actually an analysis of the pore region based on amino acid sequence, and you can create a little tree, just like a cladistic tree in evolutionary biology. You can actually put different channels together based on their similarities. So here are the sodium voltage-gated sodium, voltage-gated calcium. These are most similar. Then you have all of those trip channels, and then you have this whole big family of potassium channels. So here's the huge variety of these potassium channels. Here are the largest group are these voltage-gated channels, which are two-pore, calcium activated channels. So you really have this whole family of channels. I'm not going to ask you to memorize this particular tree. It's just to sort of illustrate how these things kind of fit together. Okay, so let's talk about structure and some of the really critical features of an ion channel. So ion selectivity, I think this will sort of be the last topic of the day, and we'll continue on next time. So the ion selectivity is critically important as determined by what's called a selectivity filter in the pore region. Okay, and then we also have voltage dependence that we'll talk about at the next lecture. Okay, so previously you could kind of guess as to where these pore regions are and where the voltage sensors are, but recently, relatively recently, 1998, it's recent for me, it's ancient history for you, really provided a structural view of the potassium channel. This is a particular kind of potassium channel that's found in bacteria, but the same principles apply. So here's sort of a side view. I presume you've all sort of seen these sort of helical ribbon diagrams before in cell biology. So this is sort of a side view. This is a top view. You have the 4 different subunits, and then the potassium ion sort of fits in the middle here. Okay, so here's sort of a cartoon version. In the center, you have this pore. You have these helices that have actually a negative charge to them. So this negatively charged pore helix, so it's kind of pulling the ion through. And the selectivity really comes from the size and the way the charges are arranged in this pore. And this is actually some kind of hardcore physical chemistry that we're not going to get into in great detail, partly because it's a little outside of my wheelhouse. But the basic idea is that you have to have these walls at exactly the right dimension in order to fit a potassium ion, and nothing smaller and nothing bigger. Nothing bigger, obviously, because it won't fit. Nothing smaller, because there's a certain way that the ion is being stabilized in the protein to allow it to move through. Okay, and I had a little video. So let's watch the little video, and then I will let you go. We'll talk about voltage sensors next. Google is really your friend. It's actually remarkably accurate, and I use it all the time.